So we are interested to understand the mechanisms that generate antibodies, and these are unique mechanisms of editing the genome. They're called rearrangement of V, D, and J segments that generate a repertoire of antibodies in the bone marrow, and this is followed by somatic mutations which are introduced into the immunoglobulin genes during the immune response in the germinal centers. And so we are interested to understand how this contributes to make good antibodies that can defend our body from infectious diseases. So what we have developed is a method to isolate human monoclonal antibodies from memory B cells or plasma cells. We sequence these antibodies, and we aim at understanding the basis, the structural basis for their specificity. So as an example, we have isolated antibodies that can protect from all influenza viruses. Single antibodies can protect from different viruses. And we also use this system to identify antigens in complex pathogens. For instance, using this approach, we have identified unusually potent cytomegalovirus neutralizing antibodies, and through these antibodies, which are now developed in clinical trials, we have been able to identify the target antigen, which is a pentamer, and make this antigen as a very promising vaccine. So this is a process by which we can use the immune response to understand which are the right targets and then make the right targets into a vaccine. Along this line, we are interested to determine how many mutations are required to make a good antibody. And I would say the good news is that very often the antibodies have already high affinity even before acquiring mutations, and often a single mutation is sufficient to make a high affinity antibodies. However, mutations continue to accumulate, and they generate a repertoire of different antibodies that can then be selected by different viruses, and in this way the specificity of the antibody can be broadened. And this is a kind of a new mechanism that is at the basis of the generation of these very useful so-called broadly neutralizing antibodies. And then in the last part of my talk, I spoke about a totally new and quite surprising mechanism of generation of antibodies. And these are very unusual antibodies that we discovered in patients infected by the malaria parasite. And these antibodies are generated actually by transposition of a piece of DNA from another chromosome, in this case chromosome 19, into the chromosome 14 where the immunoglobulin genes are. And this large piece of DNA encodes for a domain, a whole protein domain that is mutated in the antibody and gives rise to a very good binding domain for the malaria parasites. So we don't know how frequently this mechanism is used. It certainly can contribute to malaria immunity, and I was arguing that perhaps this can be maybe the tip of the iceberg, and many other pieces of DNA may be kind of stolen from other chromosomes and put into the immunoglobulin genes. So this was about my talk.